Hi, Miss Stark. Okay, so here is the close-up of a DNA strand. Here's the 5' end, and here's the 3' end. It goes from this direction. It also goes this direction on that way, so this one is flipped, and they're anti-parallel. This one has 2 bonds, and so does this, and this one has 3s because there's cats in the garden. This one is a deoxyribonucleic acid because it has 3 phosphate, a 5-carbon sugar, and a nitrogenous base. This is a phosphate, this is a 5-carbon. This is our model of DNA in progress. Here's Kenna and Bailey. Yeah, so it 5' goes this way, and at this end of the 5' it becomes a 3', and on this one it's the opposite. Here's the topoisomer, that thing. It's this, right? And so that makes it straight, and then that lets the helicase unzip it so that they go this way. The direction of the strands is that way, I think. And this is the replication fork, right? These little orange things are the SSS proteins. SSB proteins. Whoops. Yeah, and so this one is the leading strand, which is the easier one to understand, so I'll start by explaining that. The polymerase replicates it replicates that. And then so on this one, that's the lagging strand. That one's different because it has to use the primase to like line up these things, the primers, right? The DNA primers, and that's where the origin is as well. And then the Okazaki fragments, they kind of like line up and try and catch up. That's why it's lagging. And then there's another polymerase, and then it's sick, and then that's replication bubble. Bye.